But this time we added a drug called naloxone. Now this is a drug which binds to the endorphin receptors and blocks them from getting onto the cell. So basically, the idea is if you inject naloxone, it'll come into the system and get right onto that cell, get on the receptor, and prevent any endorphin from acting. So we injected naloxone, repeated the same experiment, and this time acupuncture did not work. So that was really the beginning. We then had to do a lot of experiments to verify the hypothesis. Now it's well known in science that to disprove a theory only takes one experiment, but to prove a theory takes 10 or 20 experiments, because it's always, you have to cover all the criticisms, and especially if it's in a controversial field, then you've got to do 30 or 40 experiments because nobody believes you even if you've proven it. So we have spent the next 5 years and 20 papers since the first one to try to find out exactly, A, does acupuncture work by the endorphin system, and B, what are some of the pathways and nerve mechanisms that work via these endorphins. In the previous story, you saw us recording from this cell. This is a diagram of a pain pathway. The spinal cord is here and the brain is there. This is very schematic, obviously. And the pain message would be come in here, activate a nerve, and cause something we call nociception, which is the same thing as pain. This fiber would synapse in the spinal cord, and this would then send the message up to the brain in the thalamus, where another pain cell would send the message higher up. So this is a pain transmission line. And the previous slide showed us recording from this cell extracellularly. But we gave acupuncture at several segments away. Often, if the pain was coming in the leg, we would give acupuncture in the hand, let's say. So this was far away. And what we were doing is activating deep muscle receptors, which sent the message into the spinal cord, and through another pathway, would send the message either up to the brain or through a multiple segmental path would feed back onto the cell that we were studying, the pain cell. Now, our current working hypothesis is that acupuncture activates 3 different possible systems. One is this, shown by this dotted line, which works through local segmental arcs, all staying within the spinal cord. Then there's 2 others. One that goes up through the midbrain and acts on a cell in the midbrain called the periaqueductal gray. Periaqueductal gray. This is a cell labeled P. And this P cell has as its transmitter enkephalin. So enkephalin, EK, is located right here. So the P cell, through a chain of actions, eventually activates this R cell, or raphe. R means raphe. So that acupuncture comes up, activates the raphe cell, which then sends a message down to the spinal cord in a tract called the DLF, or dorsolateral fasciculus, and releases a transmitter here called serotonin, or 5-hydroxytryptamine. Minus in this diagram means inhibition, whereas say plus means excitation. So you can see that serotonin release will inhibit the firing of this pain cell. Just like we just had endorphin coming up here. The transmitter on this dotted line is endorphin. The transmitter on this solid line here is serotonin. But either one of them can cause a negative or inhibition of that pain cell. So when we gave naloxone in our cat experiment, while recording from this cell, we think that the naloxone was blocking 2 things. It was blocking this dotted line, preventing it from inhibiting directly in the segment, and it was also acting on this system here by blocking the enkephalin from being released at this point.